Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. I'm incredibly excited to be here today with Lon Cardon, PhD, who is the president and CEO of the Jackson Laboratory. Lon has had an incredible career spanning academia and industry. Prior to joining the Jackson Laboratory, he was the chief scientific officer at BioMarin. Prior to that, he spent nearly 10 years at GSK working in target sciences and discovery. And before that, he was a professor at the University of Washington. Lon has been, and I'm going to stop there because we're going to cover some of the early years in a moment, but Lon has been behind some of the most transformational research in the genomics field in his career. So I'm really excited to talk about what he was focused on in the early days of his research career, but then towards the end of the episode, we're going to turn towards what the focus is today and what the future might look like. So Lon, thank you again for joining us.
Hey, it's a great pleasure to be here. Thanks, Patrick.
I want to start with your involvement in some of the very earliest, and I think the first genome-wide association study. Maybe you can take us back to what got you interested in the field of large-scale genetics in the first place, and a little bit about the kinds of, programs you were involved in leading and what the major questions were of the day when you were starting in this field.
Yeah, sure. So I think I'll try not to bore the audience here and go too far back in history, but it is useful, I think, to remind people or to let people know before the genome-wide association era, that before those studies were really on the scene, association studies in genetics had a terrible name. Virtually all of what we published, the community published, was questionable, and a lot of it were false positives. And there were a lot of reasons for that. Small sample sizes, we didn't have much in the way of markers, our technologies were imprecise and often error-prone and so on. But that takes us into the 1990s, late into the 90s, and about the time of the human genome sequencing, the first report of the human genome sequence in 2001, we were still pretty much just waffling around in association studies. And what came on the scene was the accessibility of larger sample sizes, better markers, and a recognition of a need for statistical rigor in what we called significant. And when we brought those pieces together throughout the early 2000s, out came genome-wide association studies. It was kind of a merger of epidemiology, technology, and genetics, and medicine.
And at the time, was that a controversial idea, or was it, that everyone understood that it needed to go that way, but you didn't have the data or the statistical methods or something to really do it, so everyone was stuck in kind of candidate gene land for a decade?
Definitely, they were stuck in candidate gene land and some. I guess we're finally past that now, but some persisted long after genome-wide association studies.
Yeah.
I don't think anyone argued that the emergence of more dense and better genetic markers was a good thing. Everyone agreed with that. I think people tended to like large sample sizes, although what was controversial was what controls to use. I know in the Wellcome Trust Case Control Consortium, we were amongst the first to use other cases, collections of other cases as controls for one disease. Boy, that was very controversial in the field at the time. And my recollection of this era was it was a magical time from about 2001 when we were getting funded for it to 2007, 2006, 2007 when they all started really coming together. The hardest part wasn't actually the science, I think, or the methodology. The hardest part was getting people to work together. And some disciplines jumped right at it. Type 2 diabetes with Francis Collins in the US, they sort of coalesced very quickly. And other communities just took years and years well beyond that into the maybe 5 or 6 or 7 or 10 years ago now only to really get their act together and realize that they will find a lot more by sharing their data. It was a big realization at the time. And I think the correlation this is anecdotal, but correlation is very strong with the emergence of real findings between that and the fact of those groups that actually work closely together.
Yeah, interesting. What do you think has driven that? Is it leadership and individual personalities and the structure of those research communities at the time that it happened, or is it something different that affected which groups found this religion sooner than others?
It's human nature, I think, right? And some groups just naturally coalesced probably because of leadership, and they realized that the data and the results that they were putting out at that time were not robust,
Yeah.
and they needed to try something else. But remember, there was a biotech wave in the 1990s. It was the first one in my lifetime, but I think there was one before that in the Genentech era, where there were a lot of people, and this is going to sound flippant, but it's not far off from this, that thought if we could just find the genes that cause common disease or the mutations or the variants that cause common disease, that drug discovery thing is easy. And that was the mentality. There was a lot of venture money
Right.
poured in, and there were companies, and so it was still a negative connotation to go from academia to industry, but no one minded protecting intellectual property in academia. That was the de rigueur thing to do, is I'm going to find those variants or those mutations, and I'm going to protect them. So that was kind of opposite of sharing.
Yeah.
And a lot of groups just didn't get over that for the longest time. And when they finally did, whoops, come all the results.
Yeah, that's interesting. That has shifted a huge amount in the last couple of decades, right? To a much more open, and you see that, like, it's amazing to me to watch what happens around the UK Biobank when data releases come out, and how just the sheer volume of amazing things that come out within typically the space of 3 to 6 months of people getting their hands on the data. The most and there's a lesson in there about how we're constrained by the number of smart brains we can point at the data most of the time, right? Not by many other things.
No, I think you're absolutely right on that. And the more smart brains that we can point and bring to bear, especially from other disciplines that have a lot of new perspectives, it just freshens everything that we work on.
You mentioned transitions from academia to industry. When you made the transition into GSK, it was very much not in vogue in the way that it seems to be in vogue today for a lot of academic folks to come into industry. Why did you make the decision at that time? And tell me a little bit about what genetics-driven target discovery or the work you're doing at GSK was like at that time.
Yeah, I actually did it twice. GSK was my second round on that.
Oh, interesting.
So I went to graduate school in the Institute for Behavior Genetics in Boulder, in Colorado. And then I went to the math department at Stanford to do my postdoc. And it was more bioinformatics, but it was with a probabilist named Sam Carlin in the math department there. And that was during so that was the 1990s, and that was during that first biotech wave.
Right.
And it really looked so exciting that if we could just get our hands on samples and markers, that this whole genetics thing was going to revolutionize medicine.
Yeah.
And I jumped right off the bat to a biotech company in San Diego called Sequana Therapeutics. And that's all they did, was just genetics. So I was there for about 4 years, and that was really controversial to go. I lost a lot of ties academically with that one. And it turned out, though, that little biotech company was well before its time, and you could see pretty early that this wasn't going to be as easy as everybody thought it was. But the founders many of them were British. And the early scientists were British, and that's what got me to the UK to go back to academia. And
Right.
And the Wellcome Trust Center for Human Genetics was just being formed, and it looked just like this company, but in an academic setting in Oxford. And so it was a natural transition for me, and so I went there and did more of the same in terms of association studies, pedigree studies, linkage type studies. But when we all came up with genome-wide association studies in 2006, 2007, or your Gott's paper a couple of years earlier. To me, that was almost existential, because we knew how to find the genes, or at least the variants, that cause or that are associated with common disease. We knew that recipe right off the bat, and that bigger samples and denser markers, and we had the kind of recipe for it. And my personal phenotype is I didn't want to just keep doing more of the same. I was immediately on to the next question, which was, all right, what are we going to do with all this? We're going to have all these loci that are pouring out. The Wellcome Trust Case Control Consortium only had about 20 in the first instance, but you knew there were going to be hundreds. I didn't know there would be tens of thousands more, but you knew there were many more coming. What are we going to do with it? And no one was really chasing that. So I was actually trying to be a step ahead by leaving academia and going to Glaxo. And at the time, GSK had the biggest genetics department in the world. bigger than the Broad at the time, I think. There were 600 people at
Huh.
GSK, early 2000s.
Why was that? Why were they so early in investing in this?
They were early adopters in genetics. There was a late Alzheimer's researcher called Alan Roses, who was a strong voice of genetics, and he helped build it up. And there was the on the Glaxo side, so Glaxo and SmithKline merged. Alan Roses was brought in from Glaxo, and there was a terrific former professor of genetics at Cambridge, Peter Goodfellow, was on the SmithKline side, and those two companies merged, and so they had two real powerhouses of genetics that managed to convince leadership to invest heavily. They were ahead of their time, but it was really quite a statement for GSK to go as far as they did.
Yeah, interesting. Very cool. You mentioned this really important fact that emerged that I think you probably didn't expect prior to the Wellcome Trust Case Control Consortium, but over the last 5 to 10 years, it's very clearly emerged that there's not hundreds of markers, there's not even thousands, but for many common diseases, there's tens of thousands or hundreds of thousands. How has that changed the way you think about the role that genetics plays in drug discovery? You've had, certainly confronted with this while you were at GSK, but I think also probably at BioMarin. What do you think about the role that it plays beyond, the simple kind of monogenic insights that we have in moving into this incredibly polygenic signal we get from most diseases?
It's a great question, and I don't think I would have expected. I know I wouldn't have expected the sheer volume of loci that are out there. And I know early on, those of us that were right at the sort of cutting edge of genome-wide association studies, some of the monogenic people blamed us for there being too many loci. Right. Like we dictated biology. That wasn't us. We just helped find them. Genetics is surprised in an elegant sort of way. It surprised me every step of the way. I really did think we would follow the monogenic path and we'd find some smoking guns that would help us do targeted drug discovery. That hasn't come to be. Who knew? I wasn't amongst them, but there were very strong voices in the genetics community that still called it junk DNA and dark matter. And all that is so fascinating and interesting now, all the non-coding regions. Where it's coming to now, I get frustrated with it, actually. Till the day I die, I'm going to push to maximize the potential of human genetics. I still think it has all these gifts to give us in terms of better therapeutics and better understanding of disease and diagnostics and so on, but not in the ways we thought. So maybe this is jumping a little bit ahead, but one of the learnings that it took me a long time to get here, but it's changed my last 5 or 7 year trajectory, and I think it's building every single day, is when I was at GSK, so as you do in big pharma, you get broader and broader and you take more and more responsibility and you get further and further away from science. And I was getting further and further away from genetics, which is really my love. And while I was doing that, a lot of things were happening in the field, and rare diseases in particular were getting better and better characterized, not just from the gene discovery side, but we were understanding how to make drugs for those and how to do clinical trials and measure the effect sizes, which often can be so much larger and so on. And we started seeing things where the same genes, maybe not the same variants, but the same genes responsible for rare diseases were actually implicated in common diseases. And that sounds really trite and sounds like precision medicine and so on, but it's actually really, really important, because in some instances, you could take and build a drug for a rare disease, which may be financially toxic, but the patients still need it. And it may be relevant for a broader segment of the population. And if you'll allow me, I'd love to give us a story on this. And this is the one that really, there were several preceding examples of this, but the one that drove it home for me was when I showed up at BioMarin in 2017. There was an ongoing program for achondroplasia, for dwarfism. It's very interesting genetics of achondroplasia. And the target that they had chosen for this was something called C-natriuretic peptide. I won't go through the biology of how it works right now, but they were into the clinic already, and it looked really, really good for adding height, helping the growth in achondroplasia. But very, very interestingly, in that same target, the C-natriuretic peptide, CNP, there were people out there walking around, potentially in UK Biobank. In fact, I think there are some in the UK Biobank that are missing that gene altogether.
Right.
And those people have 4 or 5 standard deviations below the mean in height. And there are also some people with an extra copy, and they're 3 or 4 standard deviations above the mean.
Wow.
Has nothing to do with achondroplasia at all. It's just height. And height, so to me, this is really interesting because height is it's got to be about the most polygenic of all. There's tens of thousands. I think it's 12,000 last time I saw. You can draw a Gaussian distribution perfectly by just plotting the loci for height.
Right.
And so how would you ever know where to start for a gene or a drug target for height? You wouldn't. I mean, it just got a whole distribution that you would never know where to start in there. But if you start with this one, because it's out on the extreme for achondroplasia, it'll work in anybody. It'll work in anybody with a growth plate. And so BioMarin built this drug for achondroplasia. It's approved. It's doing great. And what are they doing? They're immediately going into other just idiopathic short stature. Have nothing to do with achondroplasia because the genetics told them to. So you start with a rare disease, but you can have an impact on a much broader audience. And now we're starting to see that with epilepsies. We see it with the Gaucher disease gene, GBA, has a very strong implications in Parkinson's.
Yeah.
I was meeting with a group this morning that is really talking about glycogen storage disorders implicating Alzheimer's. Who would have thought an ultra-rare disease and Alzheimer's would be talked about in the same sentence? None of us, right? You got ultimate polygenic and rare disease. That's, to me, transformational. Maybe that's where genetics has its niche.
Yeah, amazing. You also had me thinking about Alzheimer's and some of the very rare variants that result in either extremely early onset or protective, the Christchurch variant. There's been a series of papers recently about that. And that example sprung to mind to me as well of studying these ultra rare cases, actually just being a window into the biology. It raises the point you made earlier of, in fact, genetics hasn't completely solved drug discovery. It requires, if anything, even more smart medical brains and chemistry people to figure out what do we actually do with that information. But it does provide often a very unique insight into actually what is going on through all this complexity.
It really does. And my current challenge at the moment, and I don't know how to tackle this one, and maybe your emphasis on multiple brains is the right one, and we just bring in more. We have absolutely exquisite tools genetically now. Who would have thought we would have CRISPR so many years ago, or the means to intervene with gene therapies the way that we do right now in the sequence that we can in single cell biology? We've got the ultimate microscope on the genetic side, and yet we're still trying to predict these clumsy phenotypes that we think of clinically that have been derived 50 years ago. I think genetics is becoming a phenotype problem more than a genotype problem. And that's a really tough one to crack because we think of Alzheimer's. What is Alzheimer's? Alzheimer's is probably 100 different things. Yeah. And we glue them all together in some amalgam and then try to pick them apart. Well, the genetics are telling us the truth. It's disentangling the phenotype side that we're, I think, the challenge in the future.
Yeah, you've said something really, to me, very thought-provoking previously, just about our understanding, maybe at a very fundamental level, the taxonomy of disease is wrong. And an example I've heard you use before is cancer. And we kind of have seen this play out in the genetics of cancer over the past couple of decades, that it's very clearly more useful in many cases to understand it in terms of its molecular drivers rather than the tissue of origin. Maybe you can talk a little bit more about that paradigm shift that I think you think that we need to make.
Well, so again, this is on that same thread of my realization at BioMarin that there is a continuum from rare to subsets of what we think of as common diseases. When you think a little bit about that further, we should have known that. We really should have known that, and maybe some did. But it was a little bit more than 50 years ago than the president of the US, Richard Nixon, declared the war on cancer. And at that time, cancer was breast cancer or lung cancer. That was it. That was the type of cancer or whatever. And now there are something like 80 different histological subtypes of breast cancer and 20 different lung cancer types. It broke into a bunch of different pieces. It's not a lung cancer. It's not a breast cancer. It's different types of cancer with different treatment paradigms. Why would chronic diseases be any different? Why would we? So why are we still thinking of them just as Alzheimer's as we did lung cancer 50 years ago? It just seems what we really need to do. And I picked up this phrase from NHGRI, from Eric Green's Institute at the National NHGRI at NIH, on redefining the taxonomy of human disease. And that's what they were saying. let's think about these things differently. Let's think about type 2 diabetes that's not type 2 diabetes anymore, or Alzheimer's or schizophrenia or whatever. We've got all this exquisite genetic information. Let's move beyond that. And I think that's a really good plan for the future of the field.
I saw of this, like a mini version of this happen in the field of previously known as NASH, now MASH, metabolic associated steatohepatitis. And I just thought it was really interesting actually that a field came together and said, we've got this whole naming convention and the way we organize this disease wrong. Let's come together and actually reconfigure it, rename it. And I'm interested to see how many of those evolutions different fields go through. Is there going to be a point in Alzheimer's where they say, actually, we need to break these puzzle pieces off and reconfigure this in a way that just makes more sense in light of all the findings? And there's kind of a macro level scientific process and medical process that has to go on there that you've got to imagine in most cases happens a decade too late, just because people can't organize around changing the way they think and changing the way what all the textbooks say, what do all the pathways in the healthcare system say. So it's a really important thing, but at the same time, it feels like moving an aircraft carrier in in the best of times.
It's back to the people, right? Just like we saw with GWAS, who is going to be willing to say, you know what we were doing didn't work, and maybe all the attention on ApoE and Alzheimer's is not deserving of all the attention it gets. Let's move on to something else. That is what it will take. I think it's worth mentioning a societal benefit here too, though. If this is a broad phenomenon where we can look at rare or even ultra rare diseases where there may be only a couple of hundred or even fewer cases in the world, no one's going to study those or build drugs for them from a commercial perspective, because there's just no financial bedrock for them to do that. But if we could convince ourselves that there is a path at the end, then maybe we solve a societal, a socioeconomic problem of how do we get at the 9,000 rare diseases that are too rare to be financially viable, and actually progress the field of common diseases. I mean, maybe that's our future.
Yeah. There's a fascinating previous guest we had on the podcast, Michelle Werner, who's the CEO of a biotech called Alterna, and they are using a tRNA based approach to go after, they're planning on going after around 20 different liver diseases at once in essentially a basket trial that includes ultra rare all the way up to more common diseases. And it had me thinking about, it's the exact same approach that has been taken in oncology. Let's flip the model around and focus on, regardless of the tissue of origin, we're going to go after the molecular driver of this. And I think, we're just at the beginning in some ways of how we can apply this new way of thinking to drug development. I'm curious, with this idea in mind of rethinking the taxonomy of disease, what needs to change with small biotech and large pharma drug development, in your view, to really make the most of it? Because I would guess the way these organizations are structured to go after this problem isn't probably optimal for this new reality.
No, and especially big pharma are going to be very reluctant to go for the ultra rares. I'm beginning those dialogues with some of the senior leaders in big pharma right now, and it seems like the common response is, you're going to have to show us that link between that same gene that's responsible for the rare disease. What is the reason to believe that it is really relevant for for common disease? And I think that's a fair request on their end. I don't think the genetics are going to tell us, because just saying that they're in the same gene, there's a serious mutation that causes some rare disease, and there's a GWAS locus, that there's a lot of GWAS locus in a lot of genes. That's not really enough. I think it's going to be phenotype, and phenotype might be molecular phenotype, it might be transcripts, it might be something else that links what we think of as the common disease, or a subset of people with the common disease, with that rare. And in fact, that's what drove me to the Jackson lab, was that sort of realization. We've got to go on this search for the linkages. What are the phenotypes? And phenotype, I mean, just very broadly, just about everything that's not genotype in this case, that can help us see that continuum. And that's a bit fuzzy, but right now we're doing it opportunistically. And GLP-1s didn't start being studied for obesity. They started, and we kind of find our way to these things. If we could more systematically look at that, then I think big pharma would be interested, because at the end of the day, they're very practical people. They want to go after targets that have the highest probability of success.
Tell us a little bit about Jackson Lab. It is a historic organization, one of the prime movers in the field of genetics and precision medicine. You made the decision to join in November 2021, so just about 3 years ago. Talk a little bit about the way that the organization has evolved over the years and also what drew you there and what you've been focused on.
Yeah, thanks for that. I'm really proud to be there. It's kind of a historic entity in the field of genetics. Jackson Lab started in 1929 up in Bar Harbor, Maine, and they really pioneered the use of mice to study initially human cancers. And so have built up and have built into from 1929, where the initial researchers literally lived on an island in Maine in tents. They had students that preceded the formal formation of the Jackson Lab that we just celebrated our 100-year anniversary of having students, high school and college students, spend their summers up there in tents studying genetics way back before we knew any of this. And now the Jackson Lab is 3,000 people. We provide and empower genetics research for 2,400 institutions in 68 countries around the world. We're connected just about everywhere. I can't say this with absolute certainty because I don't know, but I would surmise, because the Jackson Lab led the use of mice for studying humans as models for humans, I would surmise that just about every drug, if not every drug ever approved that has used an animal, has used a Jackson Lab mouse or a derivative thereof, which is so the impact has been phenomenal. And because mice are so useful for single gene disorders, to breed them, or now with CRISPR, with gene editing and other technologies, to be able to manipulate one or a small number of genes and then ask what happens biologically in a whole organism, they're very, very useful for rare diseases. So now, as I said, Jackson Lab, 3,000 people. We have 12 or 13 sites around the world. Our research sites are now in Connecticut and still up in Maine, and a lot of other sites for doing that. So I was really pleased to go because we have such a body of data using model organisms, and we have emergence emergence in the field of electronic health records, things like UK Biobank and all the other large resources. If we can use that model organism data, which we can manipulate, and link that to the big databases, which we can't manipulate, but we have the clinical information, maybe therein lies some of the links between the phenotypic profiles that we need to put together. Yeah. So that's what drew me to the Jackson Lab. It's this hugely trusted, beautiful place with a great history in genetics, and maybe we can have a real impact by linking up with the great genetics resources that are out there today.
Amazing. I actually, when I was an undergraduate, I applied to, I don't know if the summer program is still going, but I applied to the Jackson Lab summer program. I didn't get accepted. I was looking forward to kayaking around in between writing code and working on mice projects, but I ended up actually going to Oxford to do a rotation in a lab over there for the summer. But I remember just like learning about it at the time and finding it to be pretty amazing. The history that you mentioned, and also just the role that it's played in the field is pretty astounding.
Well, I couldn't say Oxford was a bad place since I spent some time there either, but no, the program is very much still going strong this year. Again, we just had our 100th anniversary. We had 38 students that come up for 10 weeks in the program. We had 500 applicants for 38 students. So wow. It's a great program. And it's the summer up at the Jackson Lab in Bar Harbor, Maine is quite magical because we've also been teaching the Victor McCusick short course on genetics for something like 67 years. And so what happens is kind of the good and the great of genetics come up and spend some time teaching and interacting, and they might stay for only a couple of days or they might stay for a couple of weeks. And it's just a real special time to have the latest and greatest working through. I know when I started at Jax, I think it was my first year, it was still Zoom, but the final speaker, the pinnacle speaker was Svante Pabo. And something like 2 weeks later, he was notified that he won the Nobel Prize.
Amazing.
So that's pretty cool to be able to have that level coming through.
That is very cool. You all launched relatively recently what I think is a very cool new program focused on precision medicine and cancer, the Maine Cancer Genomics Initiative. I wonder if you could talk a little bit about that, because I think it also signals a little bit of what the focus is going to be on the future of what you're doing at Jackson Lab more broadly. But I wonder if you could talk a little bit about that program, and then also what plans you have for the future to continue to evolve this amazing legacy.
I had nothing. I can take no credit for this program, but it's elegant and beautiful, and it fits perfectly. So it started out of a need in Maine. So Maine is very rural, and healthcare is dispersed, of course. And Maine doesn't have Mass General, or it doesn't, have the research hospitals that other places have. But that doesn't mean it has any less need for cancer diagnosis and treatment. And at the same time, I had someone look this up not long ago. If you ask how many clinical trials are ongoing in cancer today globally, something like 20,000
Uh-huh.
20,000 clinical trials, I think 7,000 or so in the US alone. So if you take that as more or less correct, how could any physician know what's really going on? How could any physician see a patient, get their genomic profile, and say, there's a trial ongoing in Iowa or in the UK that's exactly studying what you are facing? You can't. It's just not available to anyone, and so you can't blame the physicians on that. It's just the field is moving too fast. What was done here by my predecessor at the Jackson Lab, by Ed Liu, and now run by someone, Dr. Jens Reuter, is they got together all the physicians in all of Maine, all the oncologists come together and review the cancer cases on a regular basis. They send the samples out, they get the genomic tumors, and they have a genomic tumor board sit and review and scan the entire literature for what trials are going where, then give that information back to the primary care physician, the oncologists. They don't have to steer them, but they often do.
Wow.
To whatever trials. And the drug companies like it because then they're getting patients with the right profile for their studies. And so this is a real world trial. This is not a clinical trial that's controlled in any given way. And the team just analyzed the first set of data after 3 or 4 years of it. And those survival curves are separate. They are separating. Those people that are enrolling in these are having something like 31% or something, but that's as real world uncontrolled as you can believe. And so it's because in Maine, all those physicians will talk to one another. They're not competing, they're working together. And I think it really shows community medicine and how that comes together to tackle a huge precision medicine problem and bring the latest and greatest technology in terms of genomics together with a real practical need in the community. So we want to take this further, right? I don't know that it will work in Boston or in San Francisco or in other places where people have their own research programs and agendas and so on, but it might in other places that need it.
Yeah.
So we're really proud of that.
Yeah, it's amazing. It seems like the kind of model that would work really well in countries that are small, such that they're not the first destination for clinical trials for pharma. But if they could be coordinated and really present like a great offering, because I think of these, especially if you're showing that the curves are separating for nation states that are the size of for countries that are the size of a state in the US, making themselves an attractive place to run these precision medicine studies by having a coordinated national scale effort. I have got to imagine that this makes Maine a much more attractive target for putting a clinical trial site if people know that there is a statewide coordinated effort to say, you can actually travel to Bar Harbor or somewhere else to take part in this versus you've got to get on a plane and go to Iowa. So I'm also really excited about this. I think it's a model that could scale by state, but also to smaller countries.
Yeah, I think we haven't gone to the clinical trials yet. That's definitely on our radar. But you're absolutely right. If you're on the drug company side, there's not a real benefit for you to go to a small rural hospital. They might see one patient that's relevant to you, and that's a lot of money to go there. But if you just collapse them together from whether it's a nation state or states in the US, then you get the benefit as you would from a big urban community. So really exciting, I think, and I hope people take notice of it because it's a win for everyone, for the patients, for the system, for the drug companies.
Yeah. What have you learned about running a global organization? You have a footprint not just in Maine, but I think you're in China, you're in you're around the globe. And you've also obviously worked across the pond, both in the UK as well as the US. What have you learned about global collaboration in science?
Yeah, I was really fortunate because I did work across the pond in the UK, and I think that was such a collaborative community there that it was within the UK, but it was then it was European Union at that time when I was there, and so I learned the power of diversity and the strength of, as you put it a couple of times on this call, bringing the different brains together. And then when I was at GlaxoSmithKline, it was the same because it's multinational and everything is multinational. So that part of the Jackson Lab is not a challenge, and I really enjoy having the different sites and working under the different models. I think working in multinationals is just like working across states or even across different parts of the UK. You got to have good leadership. It's down to the people. It's good leadership. It's good principles. It's good from a corporate perspective or from a standardization perspective. I think just good procedures that can be lifted and shifted across countries.
Yeah. As we wrap up here, if you were advising someone early in their career around a topic or a field or a technology or something that if you were to make a bet right now to say, I'd bet my career going into this area that it's going to be really impactful, it's going to be really exciting, it's going to continue to evolve. I think, you made a series of great decisions in retrospect over your career on where to focus, where it was early enough that you could really, it wasn't obvious to everyone that it was the right thing to do, but you make the leap and make that decision. So I'm curious if there are any areas of the field today that you look at and say, I think this is a big part of the future and I would be looking seriously at it.
Yeah, I think from the biological perspective, I think it's looking at common diseases from this, as we discussed before, from this lens of rare. That won't be the only solution, but that's definitely real and prime time. And there's a lot of room, and we need all those brains together there. From a technology, I'll go on the simple side. Every genomics technology that we've come across, whether it's even for drug discovery, monoclonal antibodies, gene therapy, PCR, maybe not so much. They all go through the hype cycle, right? We think they're going to transform the field, and then they fall down a little bit, and then they come back and they work, but it takes a lot longer. I think editing is the one that might break that mold a little bit. And because it transforms the way we think about problems. I mean, it was my lifetime, and within my lifetime, when we wanted to study a disease, we had to wait until a patient showed up somewhere on the planet with a mutation that was of interest. And then we go, chase down and find their family members and trace the pedigree. Now we just, any undergraduate does it in a dish. And, I think it will take a little bit longer to really make that as bulletproof as possible for therapeutic applications, but still the implications and that toolbox is so broad. It's really cracked it open for us.
Yeah, it's a really interesting insight that some things go through the hype cycle, but some they crash right through it, right? And they actually, I think this debate is going on right now with AI. Is it going to deliver on all the promises because the expectations are high, or is it, are these models just going to get better and better at the pace they have been? And anyone who's betting on a hype cycle dip is actually just going to miss the whole thing. And I don't know the answer, but it's editing is a really interesting one as well, that it may just continue to surpass expectations.
But you bring up AI, and I think AI is going to be transformational in some areas and not in others. I mean, that's the safe bet, isn't it? But I think broadly speaking, we have three kinds of models of humans, right? We've got quantitative models, put AI in that category. We've got animal models of all different shapes and sizes, and we've got cellular models, iPSCs, organoids. I think those people that are really technically driven, bringing those three together, no single one is going to work for every problem. Yeah. But bringing them together, I think, is far greater than the sum of the parts for that. That's really exciting to me.
Yeah, there's some really interesting companies that are, I think, trying to close the loop on this by having really one of the challenges with cellular models and animal models is just the feedback loop. With a large language model, you've got all the internet's, the text and video data, but with biology, you have this hard constraint of, okay, our model makes a prediction, and then what happens in the cell line or what happens in the mouse. But there are interesting companies, I think, trying to push the envelope on this and really tighten the feedback loop between computational prediction, experimental output. And I think you all are probably doing a lot of that work. You have the scale to really do that in, animal and cellular models, right?
That's exciting for us, right? We could really shrink the time of drug discovery to really seeing clinical results, I think, if we get that right. And that I've never seen that with any other technology in my lifetime. So that's pretty exciting.
Yeah, very cool. Well, thank you. This has been as exciting of an episode as I'd hoped to, if not more. So appreciate you sharing some amazing stories and insights on the future with us.
Been a great pleasure. Thank you very much.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.